Okay, so today I will be talking about DNA replication. So here's a DNA molecule. Um, it has anti-parallel strands because the strands run in different directions. One thing I didn't say. A DNA molecule is made up of two strands. Here's the top, here's the bottom. At the top, um, on one side it's the 5' side, and on the other it runs from the 3' side, and on the bottom it's the opposite of that, so it's 3' to 5'. On the 5' side, it's there's the sugar, which is deoxyribose, and then on the 3' side, it's phosphate. For a DNA molecule, the these things here are the nitrogenous bases, which are adenine, thymine, cytosine, and guanine. The adenine and thymine, they pair together, the green and the blue, and then the cytosine and the guanine, they pair together, the red and the brown. So the process of DNA replication starts off with an enzyme called helicase, which will separate, bind with the DNA and then separate it into a replication bubble. And this is like a part of a replication bubble called a replication fork, which is like a template which creates a template for a new strand of DNA, or like a new DNA molecule. So now what happens is another molecule called DNA polymerase, or no, not DNA polymerase, primase. Yes, so this will bind, and then it will bring in an RNA primer, which has the same nitrogenous bases, except in place of adenine, it will have one called uracil. And yeah, so it'll bring in the RNA primer. Then after that, the DNA polymerase, another enzyme, will come in, and it will allow the other DNA pieces to come in, and this here is called this strand down here that has been formed is called the leading strand, because DNA polymerase only runs from the 5' to the 3' direction, or no, maybe it's the opposite of that, but anyways, yeah, so that makes this the leading strand. But then as for the strand down here at the bottom side, this becomes the lagging strand, because DNA polymerase cannot run in the direction that this one is, so it has to create the DNA in small fragments, which are called Okazaki fragments. So one fragment at a time, it'll insert the DNA. So after this happens, towards the end of the process, an enzyme called exonuclease will come in, and it will remove all the RNA fragments and just have DNA. So yeah, the DNA polymerase will come in and just put DNA in place of where the RNA fragments once were. So, for all of this, there's going to be an enzyme called topoisomerase, which just basically keeps things in line and makes sure that the DNA molecule doesn't overwind or underwind. Yeah, so at the end of the process, DNA ligase, another enzyme will come in and just clean everything up, and at the end you should have there will be two DNA molecules. Because of this, we know that DNA is semi-conservative because each DNA molecule is made up from one old strand, like as shown here, and then one new strand as shown right here. Yeah, so that is about it for the process of DNA replication.